Next, I'm excited to introduce Professor Saori Kosono as our next speaker. Professor Kosono is an associate professor in the Graduate School of Agriculture and Life Sciences and the Collaborative Research Institute for Innovative Microbiology at the University of Tokyo. Her research interests span post-translational modifications, metabolic regulation, and protein condensation. Her contributions to the field were recognized with the award for women scientists from the Japan Society for Bioscience, Biotechnology, and Agrochemistry in 2018. Please join me in welcoming Professor Kosono.
Okay. Thank you for introduction. So, first of all, I would like to thank the organizers for giving the opportunity to present our research. And my title is Catalysis-dependent condensation of citrate synthase involved in glutamate overproduction in Corynebacterium glutamicum. And Corynebacterium glutamicum is well known as an industrial producer of amino acids, including glutamate, that is umami compound. And this bacteria overproduce glutamate extracellularly in response to cell surface stimuli, including twin body addition, which induce to open the mechanosensitive channel, and glutamate is excreted from here. Under glutamate producing condition, a global increase in metabolic flux from glucose toward glutamate synthesis also occurs. And this situation is very unique because metabolic flow is concentrated mostly to glutamate synthesis at the expense of growth. And this situation is also desirable from the point of microbial production. So we are interested in the mechanism. Under glutamate producing condition, no increase in translational levels of an enzyme observed. So this suggests post-translational mechanisms is likely involved in the global increase in metabolic flux. As for the post-translational mechanism, we now focus on the protein condensation. Recent studies suggest protein condensation regulates metabolism in several situations. Protein condensation can increase the local concentration of enzyme and substrates to enhance the reaction. It also can regulate the branch point of metabolic pathways by compartmentalizing the certain enzyme. It can also separate the toxic intermediate, which results in the increasing final product formation. So we focus on the possibility this protein condensation may play a role in global increase in metabolic flux and extracellular glutamate production. By GFP tagging of enzyme involved in glutamate production, we found citrate synthase, CGCS here, forms condensates in C glutamicum cells. And citrate synthase catalyzes a reaction to condensate oxaloacetate and acetyl-CoA to produce citrate, and is positioned as an entry into the tissue cycle. From experiment revealed, these condensates had fluidity like liquid droplets formed by phase separation. And these condensates are observed both in glutamate producing and non-producing condition, like here, but several features seems different between the two conditions. For example, the size of condensates are reproducibly larger in glutamate producing condition. Using strep-tagged CS proteins, CS strains, we performed a sucrase density gradient ultracentrifugation analysis. And results found CGCS protein was reproducibly migrated in smaller molecular weight fraction in cells under glutamate producing condition compared to non-producing condition. So this also supports our idea CGCS protein had different characters between the glutamate producing and non-producing conditions. And CGCS condensates are observed in growing cells, as shown here, but not in 24-hour stationary phase cells. And Western blot analysis revealed CGCS GFP proteins were equivalent between 9-hour growing cells and 24-hour stationary phase cells. The phase when CGCS condensates are observed was corresponding to the phase of glucose consumption. So it suggests CGCS condensation is likely linked to glucose metabolism. To further characterize CGCS condensation, we constructed catalytic site deficient CS mutant strains. Yeah. And this CS mutant strain showed decreased growth and also loss of glutamate production without citrate. And external citrate addition restores growth, but still not glutamate production. So this suggests CGCS has no specific role in glutamate production, just in addition to supplying citrate to support the growth. And interestingly, this mutant CS showed a loss of condensation. We suspect the loss of condensation may be caused by the change in the environment, intracellular environment, due to the CS activity. So we co-expressed both wild type and mutant CS protein in the same cells. In the control strain, a genome-derived and plasmid-derived wild type CS protein both formed condensates. But in this strain, CS wild type formed condensates, but CS mutant did not form condensates. So this result suggests the loss of condensation depends on the protein property itself, and CGCS condensation was correlated with CS activity. To further determine the correlation between CGCS condensation and glutamate production, we determined the condensation in several glutamate production deficient strains. And we found that CGCS condensation was impaired in several glutamate production deficient strains, as shown here, and for example, lost and enlarged, and fewer and smaller. So it is interesting. This abnormal condensation was observed only specifically in glutamate producing condition, but not in non-producing condition. So this suggests loss of CGCS condensation was correlated with loss of glutamate production. And here we focus on this Pepsi deficient strain, which shows abnormally enlarged condensates. PEP carboxylase, PEP-C, is an anaplerotic enzyme to supply oxaloacetate and is essential for glutamate production. We already reported. And we have two Pepsi variants, KR and KQ, at lysine 653, which shows a moderately decreased and severely decreased Pepsi activity. And we found that abnormal CGCS condensates are correlated with a decrease in Pepsi activity. And Pepsi produce oxaloacetate, and oxaloacetate is a substrate of CS reaction. So we consider the possibility this abnormal condensate is likely due to the decrease in oxaloacetate level. So we next suspended these cells forming abnormal condensates in oxaloacetate solution and to supply oxaloacetate externally. The result is shown here. So abnormal forms in Pepsi deficient strain became returned to normal by external supply of OAA. And normal condensates in wild type became dissolute by external supply of OAA. So this result suggests oxaloacetate is the factor affecting CGCS condensation. In general, protein condensation occurs above the saturation concentration of the protein. It's called CSAT. And it can be easily assessed by turbidity measurement. So we incubated recombinant CS protein with various concentration of oxaloacetate. And the change in turbidity before and after incubation was measured. First, in the absence of oxaloacetate, a mutant protein showed lower turbidity. That means a higher solubility compared to wild-type protein at the same protein condensation. This means mutant protein had a higher CSAT value than CS wild-type. And oxaloacetate reduced the condensation, turbidity of CS wild-type protein, as shown here, in an oxaloacetate-dependent manner. But this effect was not very observed with mutant protein. And pyruvate, malate, and 2-oxoglutarate do not have such effect, so this effect is oxaloacetate specific. It is generally considered citrate synthase CS undergoes a large conformational change during the catalytic cycle. So oxaloacetate first, and then acetyl-CoA binds to the catalytic side pocket, including histidine 316, which induce a conformational change from open to closed form. And we can see this conformational change by intrinsic fluorescent measurement, as shown here. So for the for the wild-type protein, so oxaloacetate reduced the peak intensity, and also peak shift to longer wavelengths was observed in oxaloacetate-dependent manner. But such oxaloacetate changes in fluorescence profiles was smaller with mutant protein. We also estimated apparent KD value of oxaloacetate for CS protein by plotting the peak intensity against oxaloacetate concentration. And we obtained the KD value with 2 orders magnitude higher with mutant protein compared to wild-type protein. So this indicates oxaloacetate-dependent conformational change less occur with mutant protein. I here summarize the first part. Catalytic site deficient CGCS protein did not form condensates in cells. And oxaloacetate is a substrate of CS reaction affected CGCS condensation. And oxaloacetate induced conformational change of CGCS, which was fewer in the catalytic site deficient mutant protein. And based on these results, we propose CGCS condensation is catalysis-dependent, which means substrate-dependent conformational change is required for CGCS condensation. It is generally considered multivalent interaction is important for forming biomolecular condensates. In the case of E. coli LPLA, different types of dimers have different contact interface, which contribute multivalent interaction involved in higher order assembly and its protein condensation. In the case of CGCS, conformational change during the catalytic cycle would produce multiple forms that have different contact interface, which would contribute to multivalent interaction we consider. CGCS does not have a typical low complexity sequence and also does not form higher order assembly. They are often involved in phase separation or protein condensation. But this protein forms condensates. So if our hypothesis is true, this is first example of catalyst-dependent mechanism for protein condensation. In the last part, to understand the metabolic impact of loss or abnormal condensation, we performed metabolite analysis. First, in the case of CS mutant strain, which shows the loss of condensation, as shown here, in a citrate added condition, indicated by green color, to oxoglutarate, it's a precursor of glutamate synthesis. So extracellular 2-OG level was equivalent to wild type. But extracellular glutamate and also glutamine was not detected extracellularly. As for the intracellular level, again, 2-oxoglutarate level was equivalent, and intracellular level is equivalent, and also intracellular glutamate level was also equivalent. And meanwhile, intracellular glutamine and lysine were highly accumulated. So this suggests metabolism to generate 2-OG, it's a precursor of glutamate synthesis, were active. And also, intracellular amino acid profiles were fluctuated. We also determined the enzyme activity of glutamate synthesizing enzyme, that is DDH and GOGAT, and they are active. So the loss of glutamate production was not due to the decrease in glutamate synthesizing activity. Next is a case of Pepsi deficient strain, which shows this uniquely enlarged form of condensates. this enlarged CS retains activity equivalent to wild type. And GDH and GOGAT glutamate synthesizing enzyme activity also equivalent or less higher than wild type. And extracellular 2OG and intracellular 2OG level was also equivalent or rather higher in mutant in Pepsi strain, and intracellular glutamate glutamine level also equivalent, and intracellular lysine was highly accumulated, similar to the case of CS mutant strain. So this suggests again metabolism to generate 2OG was active, and intracellular amino acid profiles were changed, similar to the case of CS mutant strain. So in conclusion, we consider appropriate CS condensation is required for the distinct mechanism in extracellular glutamate production. As mentioned before, under glutamate producing condition, metabolic flow is disproportionately focused on glutamate synthesis at the expense of growth. So this is very unique metabolism, I think. And we do not know the exact mechanism, but we consider CGCS condensation has a key role in achieving the unique metabolism. And we expect understanding the mechanism underlying the unique metabolism will provide a hint for metabolic design for other useful compound production. This study was mainly performed by here, Mr. Makoto Nagaoka. He's a PhD student. And this research is performed in the biotechnology laboratory supervised by Professor Nishiyama. Thank you for your kind attention.
We have room for several questions.
Hi. Interesting talk. So you mentioned about multivalent interaction for the protein condensation. Can you elaborate a little bit more about what specific interactions that people already found? What specific interactions?
Yeah, now we are addressing that issue. So now we are trying to identify the residue, which residue involved responsible for the condensation. Yeah.
I have a question. So you saw intracellular accumulation of glutamate, but they didn't really see any extracellular glutamate. So could the condensate somehow affect the transport of glutamate from intra to extracellular?
Okay, thank you for the question. We also consider the possibility. And so we also performed metabolite analysis using the channel deficient strain. In the strain, intracellular glutamate was accumulated because of the loss of channel exported. But in our strain, in our CS mutant or Pepsi deficient strain, intracellular glutamate level was equivalent. So that means we think the problem is not export block, but maybe metabolism has problem, I think.
So you also, a follow-up question. So you also saw lysine overproduction. So could there be an indication, have you tried or do you plan to do some sort of RNA-seq or any transcription level or translational level analysis to understand why there is like a higher flux toward lysine synthesis?
Yeah. But not yet. Yes.
All right, let's thank the speaker again.
Thank you.